Hi everyone, welcome to the Board of Governors of 2020. I'm very glad for the opportunity to host you here in my lab. My name is Ido Caminer. I'm an assistant professor in the Faculty of Electrical Engineering, and we're now inside the microscopy center of the Technion. We call it MICA. It is inside the Faculty of Materials, Science and Engineering. And what I'm going to show you now is the lab that we built here recently that is combining the electron microscopes with strong lasers, both high power and short duration. And this is also why we have those double doors here for eye safety and for the quality of experiments in the lab. So we'll go inside. You can see some of what is making us really excited about this lab and what we can do with it. So what you see at the center of this lab is the electron microscope itself. This is what we call a transmission electron microscope, or a TEM. And this TEM is bringing down electrons to allow us to image samples, to take photos of samples at very, very high resolution. And the lab is really designed around it to support it in terms of the quality and stability of what we do inside. For example, we're sitting right on the rock of the Carmel Mountain, and also in other parameters like the air conditioning system here that has to be extremely high, extremely stable in its temperature and also humidity. Now the core of the system and what makes it really run is, in our case, a laser that is starting the entire process. This is really the key to everything here. This is a femtosecond laser. Those are super short pulses of laser light that is starting the light emission, and then by converting it in different ways, sending the light around the microscope and eventually into it to start the process, to excite the electrons. The most important part here is that we convert the laser light into ultraviolet. Ultraviolet, or UV, is also the range of frequencies that allow us today to kill viruses, and is really important in the era of corona. It's actually part of the research of my group to develop and invent new methods to create radiation and light sources in different regimes of the electromagnetic spectrum, UV being one of them. Here we use it to send it around the microscope and through a lot of complicated optics to couple it and then guide it through this optics. So we have two pulses eventually. The original laser pulse that is sent through the microscope here, and then the UV pulse that is sent through here. It's sent through a mirror into the top of the sample, excites an electron inside from a tip. This electron is accelerated down the column, that's reaching to about 70% the speed of light, and then they both meet the electron pulse and then the other light pulse in the sample area. Then we can put different samples inside, from photonic structures to two-dimensional materials and a lot of other quantum materials that we are looking into and studying their properties, and then collecting the information about it by looking at the electrons down at the bottom of the column, where we can collect a lot of information about electrons. For example, see tiny changes like in the absorption or emission of a single quantum of light, single photon by that electron. And that allows us a lot of the discoveries that are happening here these days. For example, the paper that appeared a few weeks ago in Nature was the first time that we were able to see how light flows on very small materials. We can actually capture light flow on a photonic crystal. Those are the same kind of materials that are used in other forms to improve the screens of our cell phones. And when we understand better how light flows inside them, we can really design them to be better. And we also combine different kinds of materials into them to improve their quality and the way they operate. And really what we saw here is, for the first time, how light flows inside such a material, being able to image it at extremely high resolution in space and also with very good resolution in time to see the dynamics of light in such a material. That's what got it into Nature. Generally building this and combining the pieces, the automation, the software, and a lot of complicated science, from electron physics to science of light, it's only possible because of, first, a lot of hard work by the great guys in my team, from students, postdocs, and also the lab engineer, Rafael Dehan, who's really the father of this system. And also, what made it possible for us to start here is the support of the Technion administration, friends from abroad, the Magid family that supported this financially and made it possible. And for us, Technion is really an accelerator. I came with this idea, built it originally before I came back from MIT, and the fact that Technion supported this with the ability to put funds into it, the space, the area, the expertise of different people is really what made this possible. So for me, this is an accelerator for the scientific startup that is now leading to a lot of beautiful science, and we're doing more of it these days, and I think we'll see more exciting results. This is a lot of fun for guys in my group. I hope to be able to also host you here physically in the lab after Corona ends, and I'm glad for the opportunity to take you for this tour. Thanks.
I'm Naama Geva-Zatorsky. I've opened my lab here at the Technion almost 3 years ago at the Rappaport Faculty of Medicine, before which I was at my postdoc at Harvard Medical School in the lab of Professor Dennis Kasper, where I was fortunate to make my dream come true, which was to study the gut bacteria, the small organisms, each one cell, that communicate between each other and complement our physiology. And here in lab, we pursue these studies, studying the mechanisms of these interactions, which microbial molecules are important for our health, and how do they communicate with us. So I'll show you my lab. This is the most important room, which is the room where we grow the bacteria. The gut bacteria are anaerobes, so they grow without oxygen. Oxygen actually kills some of them. So this is why we need to grow them in this special chamber, the anaerobic chamber, where we wash out all the oxygen, and we can grow the bacteria in different types of conditions. And here is an example of a tube where you see the media is not transparent, meaning the bacteria grew in it. Otherwise, it would have been transparent. I will also present you to my students, so we can start here first with Haitem and Niru. Haitem is studying the interactions between the gut bacteria and the brain. It's a new field which is called the gut-brain axis, and this we're doing in collaboration with Professor Asya Rolls, also here at the Faculty of Medicine. And Haitem and Niru are studying how can we fluorescently label the gut bacteria to visualize them live, both to study the interactions between them and how they complement the host. You can see bacteria in blue, in green, and in orange. And these are 3 different bacteria labeled with 3 different colors. You can see them simultaneously while they're living, and it can be also visualized inside live organisms. This is Maxime, who has recently joined the lab. And this is Rawi, who is studying several projects. Rawi is studying how the gut bacteria can be used as therapeutics for cancer, both to cancer prevention and for cancer eradication. And together with Nadav and many others in the lab, they led recently a project on corona, actually, where we developed a protocol to detect corona in people. It's actually now developed as a home kit. So pink is negative and yellow is positive. And the idea is to spit and visualize in several minutes whether you are positive or negative for the virus. Nadav and Rawi also study antibiotic-resistant bacteria. You can see here a plate with bacteria that are antibiotic-resistant. So it's a special plate where these bacteria grow black. So you can count them, and after the method that they developed, you can see that the microbes, these antibiotic-resistant microbes are eradicated. And the last bay here is the bay of Lilach, Noah, and Dana. Lilach and Noah are studying how the gut bacteria communicate with the immune system, and the actual interactions, the actual molecules, and the crosstalk between them. And Dana is studying the viruses in the gut, the bacterial viruses, so viruses that infect bacteria. How are they important to bacterial functions, and how does this affect the host? And the last room is this room, which is called the tissue culture room, where we grow bacteria together with host cells, mainly the cells from our immune system, sometimes cells from our blood, sometimes other cells. And that's how we can study the actual interactions between the bacteria and the host. So all in all, we study how these gut microbes, which are called the forgotten organ of our body, can complement our physiology. In which disease states are they important, and how can we harness them as therapeutics? I would really like to say it's very exciting to talk with you. Any of this work would have been possible without your help. It's very crucial to us. And this Technion environment is a really amazing place to work at. A lot of collaborations, a lot of expertise, engineering, biology meet together and really work together on projects. I really feel we're at the cutting edge of science worldwide. Thank you very much.
With me, my guests Naama and Ido. Naama and Ido, I would like to invite you to a glass of wine.
Thank you.
Ido.
Thanks.
And I would like to raise a toast to your health, L'chaim, for your
L'chaim.
wonderful achievements at the Technion.
You too. L'chaim.
So we got some questions here, and the first one is for you, Ido. What was your reason to come back to the Technion out of the choices you had? What convinced you to build this lab here?
Well, I hope you can hear me okay. Let's see. So I'm actually the Technion in my family. We always had a great appreciation of this place. You know, I grew up here practically in the Madatech, in the old Technion campus. My parents both were students here. My grandfather actually did Aliyah in 1946 to be a student at the Technion. So it was somewhat natural for me to be a student here. But coming as a faculty member, it's a bigger decision, I think. It's a very important junction for us in the career. And for me especially, I think it's the culture of this place that really made a difference in deciding to come here. I had those, well, let's call them wild ideas, ambitious lab to build, and I got the people here to support it, to encourage it. But more than this, I think what really made a difference for me is that I saw people delve into it. That we had scientists here that delved into those ideas and tried to make them even better. And we really see that the lab today got better because of the contribution of ideas from people in campus. And this is something that I don't think I saw anywhere else where I interviewed for positions. And people like Professor Gadi Eisenstein, lab engineers, Tony Jensinger, Professor Moti Segev, even Professor Uri Sivan, that's now the president, were really involved in trying to bring ideas to how to make this better. And this is really, this kind of spark, that's part of the culture that I liked. That's what made a difference for me.
Naama, how come we realize the existence of the microbiota only now?
It's a great question. We actually co-evolved with them since the beginning of humanity. But they're so tiny, we didn't see them. And they are actually anaerobes. They need oxygen. I mean, they need to live without oxygen, so oxygen kills them. So it's not easy to grow them. So we weren't aware of them. But about 2 decades ago, there was a huge achievement in sequencing machines that enabled us to perform deep sequencing in natural materials. So actually in stool samples, we can perform deep sequencing and realize that there are so many microbes there. And that's what opened up this field of study. Since then, we're both sequencing them and also culturing them and performing both novel, cutting-edge technology with them and also old-school technology with them, and it enabled us to study them. So we now realize their importance. They're important for our health. We acquire them when we come to the world at birth. Acquire them from the people around us, from our parents, our neighbors, our friends. And as the diversity of these microbes is bigger, we're healthier. This is a rule of thumb.
Oh, this is fascinating. Ido, I was wondering, can you actually use radiation to kill the corona, the virus?
Well, we are working in my lab on creating radiation sources, but there are actually areas in the electromagnetic spectrum we call ultraviolet that's already well known for many years, and definitely there is scientific knowledge for over 100 years of how this can be effective in disinfecting and reducing infection, and that's something that I think can help And we're looking into some other mechanisms by which light and laser light in particular can be particularly important for trying to reduce infection in public areas. So yeah, and that's a very important topic, I think, especially today.
Now, can we use those microbes later on down the road to as also medicine, as a medicine?
Yeah, it's a great question. These microbes, although they are inside our body, we think that we can use them in the future for therapeutics. So some of us lack some of these microbes, or when we, God forbid, become sick, we perhaps need some supplement of these microbes, or even perhaps more control their molecules that affect our health. So we have now probiotics on the shelf, but we believe that we need further research to understand exactly which microbes are important at exactly which health condition, and which molecules of these microbes are important. So yes, I believe that in the future our medicine will be composed of either microbial molecules or the microbes themselves. So don't be afraid of microbes.
Here's another one for you, Nama, from Joyce Silver. Are you scaling up your rapid salva test for COVID for the general population? How far from are the results?
Yeah, thanks for this question. There's actually a startup company that is starting these days with an entrepreneur that came to the Technion, and when he realized about our work, and yes, we are working on scaling this up together with this company. The name is Naor. And the entrepreneur is Oren Firth. And we believe that it will be first at clinics or drive-ins, so scaling up to perform the corona diagnostics at site, because the result is in about an hour, in even less than an hour, and doesn't require professional experience or any sophisticated machines. And further down the road, we believe it will become also a home kit, because it's very easy to use, but that requires a little bit more development. But yes, we see that it's very important and in need, and hopefully it will help the world.
Ido, how do you see the importance of advanced infrastructure like your microscope change in light of the limited international traveling in our modern corona world? That is, does your work suffer from the limitations on international collaborations?
Well, we cannot do experiments in Zoom. That doesn't work. We need to be physically in the lab to really make those things happen. And I think that the fact that we have this kind of infrastructure here, that there is the investment in making this happen here, is really important, because it means that we suffer less from corona happening. Because for many scientists, and for us as well, right, we need to travel to get to facilities that Israel doesn't necessarily have the money to build, right? That there are a lot of important facilities, synchrotron, right, that we have to travel to access, and that we cannot count on Israel always to have. And advanced electron microscopes is a good example of this. And now the fact that we can have that here means we can actually do research without being harmed by stopping us from traveling. That's the main thing that really slowed down a lot of our collaborations today and these days, and didn't hurt a lot of what we do in the lab. So I think that the fact that there is investment happened here is a big plus for being able to do research even when the world is fluctuating like crazy.
Now let me challenge both of you. I know. Let's say that I give you $1 million now, cash. What would be the next collaboration that both of you would do in kind of research? In what kind of research would you collaborate? I brought my check, my checkbook with me.
We talked a little bit. We just got to know each other, but we found actually in a couple of minutes several points of contact.
Yeah, I think that there are some quantitative tests that can be done to see how viruses, and corona as an example, suffer under radiation. And there is a lot of knowledge about this for many, many years, but nothing very quantitative. We need to really evaluate what happens when you scan over wavelengths, over colors of light, and see how it affects. What is, for example, the optimal color of light to try and kill corona? We know the sun does it already.
but can we isolate a narrow spectrum that does it without hurting us in a way? And that's something that needs research between physics and biology and medicine to be able to do.
And the second point of contact that we were talking about is to combine theory with biology, with experimental biology. I grew up in this systems biology way of thinking in Oriolan's lab, and Ido and I were talking about
like network science.
Yeah,
like
network science
algorithms for. Yeah, this is a super interesting part of mathematics that I think can contribute a lot to when coming between fields.
So in lab we see that the microbes communicate with the host, and the host communicates with the microbes, and I really see power in modeling, mathematical modeling of these interactions to predict how this will happen in different types of, this interaction will happen in different types of disease states. And I think this is something we can perhaps think about doing
together. Would you use the equipment in a lab to try to evaluate the model as you go, or how does it work?
Well, so I guess typically, but we need a little bit more time to really make it a concrete.
Yeah, I think
this will be starting from experiments that happen in the labs, and then get theory coming from more physics background.
So there's going to be a tango between theory, practice, practice, theory, and then
we see these kinds of things happening in like electrical engineering in my departments that's something that we see happening between biology and engineering, but I think there could be much more of this.
Yeah, so the model will explain what we observe and then predict what can happen if we make the experiment a little bit more complicated, and then we can test it. Or maybe the model can tell us in which condition this interaction will be most valuable, and then we can test that condition instead of scanning through thousands of conditions. So
this will be a fascinating world, and I think this is why Technion now is putting a lot of efforts in creating these hubs, research hub, and one of which is, of course, the human health, which will be a natural home for these kind of questions to ask in the future and to answer, even more important.
Agreed.
Thank you so much. Thank you so much for your
thank you very much
for the questions and answers, and thank you for everyone for asking questions. Great questions.